Okay, welcome back again. This one's just a quick video, hopefully a short one, that remind us of some of the key features of cardiac anatomy and to prepare us really for the lab, the dissection lab, in which we're going to try and truly in a hands-on manner reflect on the structures we see when we do a dissection of a heart and then what the possible functional connections might be. That's the purpose of the lab. This is just a little bit of background for that lab. For context again, we're talking about the cardiovascular system in mammals, and we're going to focus this time on the two pieces of this diagram that show the heart. And you see the left heart and the right heart are shown separated with the lungs in between them, and that's a functional or schematic explanation of the organization of the heart. Of course, in reality, it's one organ that's coupled together, but the lungs do sit in between in terms of the circulation, and we have to keep in mind this difference and this series connection between the left and the right heart as we dissect it, as we try and understand its relationships of structure and function. And here are some typical views just to give you some orientation before we start. There'll also be a plastic model around in the lab that we can use as reference. Of course, knowing that the human anatomy and the bovine anatomy could very well be different. Different species, different animals have different anatomies. We have to take that into account. The differences are not dramatic in this case, but they might be meaningful, so we have to be aware of them. So here's a picture, a frontal view of the human heart as it sits in the thorax. You can see the chambers, the labels there give you some of the idea of the vessels, but this is how the heart sits underneath the rib cage inside the human heart. At the bottom of the heart is the diaphragm, so the heart sits pretty directly on top of the diaphragm. Here's another picture of just the heart by itself, now looking at it from the right side. The key thing here is to observe the different chambers again and see the mostly right ventricle that we're seeing here and the right atrium that goes with it. The superior infra vena cava showing the path that blood takes to get back into the heart. And then you see the blood leaving the heart through the aorta. That's sort of coming out of the left side of the heart, which you can't see very well in this particular picture. We do see the coronary arteries quite well, and that's another piece of the dissection we'll have to pay attention to. Coronary arteries supply the heart with blood, so they're very important for the function of the heart. Here's another view, this time from the back. Again, human heart as seen from the back, and you can see there's a major branch of the vasculature, both the venous and the arterial side, that goes down the posterior or rear side of the heart. And what we also see is a rear view of the left atrium and that upper left corner there showing the pulmonary veins, which bring the blood back from the lungs into the left side of the heart. Again, we'll be able to view all these things from the perspective, at least, of a bovine heart. Now, if we slice open the heart, this is a little more artistic rendering than an actual photograph, but you now see the clear differences in the left and the right ventricles, the wall thickness, the shape. You also see some of the internal structures that we will see in the bovine heart, and we'll want to identify those as well. Finally, the opened left ventricle. This is just the left ventricle with a slice cut open. We're going to replicate this same sort of configuration in the lab, and you see the thick walls, you see the papillary muscles. You also see the aortic valve. So this has been carefully excised so that the aortic valve gets opened up, and we can see the individual leaflets of that particular valve, as well as the mitral valve. So this is a rendering or a view that we'll try and replicate in the lab. Finally, here's a blow-up of the aortic valve itself. This is even more close-up, so this is showing its structure. The individual little pockets is what they'll feel like when we observe them. So we'll want to open that part of the heart very carefully to preserve the valve so that we see it in as close to intact a form as we can, given that we obviously have disrupted the whole left ventricular structure to even get access to it. The right atrium and ventricle have a different look and feel to them, and those are observations that we'll want to make too. How is the left ventricle different from the right ventricle structurally, and even in terms of things like the surface texture? Here you see some red tissue, some white tissue. What does that all mean? How does that play out in an actual heart? We see the upper part here is the right ventricle, the upper part is the right atrium, lower part left ventricle, and you see them both opened up, so we can see the two different surfaces very clearly, along with some other key anatomical features that we'll try and look for in the bovine heart as well. Even a more close-up, here's the right ventricle with the tricuspid valve and the chordae tendineae. So this is showing the elements of that valve sort of seen from the bottom, and you see this highly what's called trabeculated surface structure that we'll certainly observe when we do the dissection. So all of these are elements that we should be able to see and hopefully also photograph in our dissection. This is a piece that'll be harder to photograph quite like this because in this case the conduction system, which is an electrical structure or a structure with electrical function, which we will talk about, that won't show up quite this way. This takes a certain coloring. Turns out iodine will be absorbed selectively by these fibers and produce this kind of image. We won't be able to replicate that in the lab, but you can at least appreciate the complex nature of that underlying conduction system and try and see if you can see maybe some signs of it in the cow heart as we open up the left ventricle and have a look inside. Key structurally to keep track of is the coronary veins and arteries, and here we have a diagram identifying the different arteries. We'll try and again identify as many of these as we can in the bovine heart. These coronaries are incredibly important for the function of the heart. They really enable the heart to have its own blood supply. Strangely enough, even though all the blood of the body passes through the heart, very little of it can be absorbed through the chambers. The chambers are sort of waterproof, and it takes a separate blood supply to provide what's necessary for the coronaries. And we will be able to hopefully look at them exteriorly, as you see here in the schematic diagram, and also try and find the ostia, which are the openings from which or through which the blood will flow as it leaves from the left ventricle, heading up into the aorta. We'll see two branches coming off that opening just above the aortic valve that provide the access to the coronary arteries. We'll try and identify those in the lab. So again, this is to give you just an overview of some of the different views of the heart, many of which we'll try and replicate in the lab. So come sort of prepared with these views in mind and with the goal in mind with your photographs to capture a similar set of images to describe your heart, the heart that you have to dissect in the lab itself. Thanks, and we'll look forward to seeing you soon in the lab. Bye for now.